start with the initiation process. See, this is the coding strand and template strand. Okay, the template strand. What happened now that the RP RNA polymerase doesn't initiate by itself. What happens that first it binds with the sigma factor. Now sigma factor does what, that it finds the entry point. You always remember that the RNA polymers will have to enter into the process of transcription through the entry point only. He cannot enter into from anywhere. Okay, they have to recognize the entry sequence, and then they have to enter through that point only. See, if we want to compare that with something like that, let's take an example. I have made it easier for you. That, see, this is one of the building. Okay, 1 2 3 4, it is a four. There is a fourth floor. There is 4 floor in the buildings. Okay, in a flat. And there is one thief. There is one of a thief. Okay, who came to stole one of the diamond present in the fourth floor. Okay, so what the here I have compared the RNA polymers with the thief. I have written chore, that's why. Okay, and the sigma factor is the rope, because he can't climb without anything na, so he'd want a rope. So I have compared the rope with a sigma factor. What does the thief cannot initiate the process of the stealing the diamond without the rope. So he need that rope. That means that the RNA polymerase need the sigma factor to enter through the recognized entry point and enter through it. So the RNA polymerase, what happens, first combines with the rho factor that recognizes the entry point, and helps in reaching the entry point. It takes the rho factor. What does that? The rho factor recognizes the entry point and takes the RNA polymerase to the entry point. Okay, and then after as soon as the RNA polymerase reaches the entry point, the rho factor gets out from here. Rho factor leaves the RNA polymerase, and it breaks the hydrogen bond in DNA. Does it breaking the, and does a fork like appearance is done. See, separating the two strand by breaking the hydrogen bond. Okay, so here remember that the sigma is first recognizing the starting point. It also starts the process of transcription, and also separate the DNA, and by breaking the hydrogen bond. Next step is elongation. Okay, then see, the RNA polymerase bind to the starting sequence, and then now what will do, it will start the process of elongation. See, okay. So then there is also two starting endpoints. Okay, one is the Tata box. See, Tata box, ma'am told this one about that Tata box. It is the sequence TATAAT, Tata box. It is also called the prim now. And another is minus 35 hexameric sequence. This is the sequence TTGACA. Please remember these 2 points. RNA polymerase, after binding with the sigma factor, can entry from these 2 points. Okay, it is present. See, position, you can see this is the promoter box. Obviously there will be one promoter box. See, this is the position where it is present. You have to also remember, it is called this entry point is called the prim now. Its position is the -9 or -10 nucleotide upstream and downstream. I'll talk about that later. From first transcribe nucleotide in coding stand. Okay, and the 35 hexameric sequence position is -35 nucleotide upstream from first transcribed nucleotide in coding stand. Okay, so this is the This is the position and the name of the two entry points from where the RNA polymers can enter and start the transcription process. So see, this is the first transcribed, this is the first transcribed, this is the first wait huh. So uh, uh, after reaching and binding to the, he first found from where he want to transcribe, and then he transcribed this one, is the first, the transcribed R mRNA. Okay, so this is the first transcribed nucleotide. He first transcribe this nucleotide, and then he will transcribe like this. You see, in the next step, this is his transcribing in this position, +1 +2 +3 is transcribing in this direction. Okay, so the RNA polymerase, which the direction in which he transcribed the nucleotide is called the downstream. Okay, and it is called down, or you can also state that like this is the first transcribe this one. If the RNA is moving backwards, like if it is moving towards the entry point, then it is called a downstream. But if it is moving away from the start entry point, then it is called a downstream. So this is the difference. If RNA start to transcribe in this direction, this is called the upstream. But if the RNA starts transcribing in direction, then it is called a downstream. Means downstream is the point if the RNA polymers move away from this entry point. Okay, so this is a summarization. So now RNA is transcribing new nucleotide, and thus process of elongation is continuing. Okay, so I think you have got everything in this part. Okay, the two entry point from where the sigma factor first recognizes the sequence and binds to the DNA sequence to initiate the process. Okay, so in the next step what happens, see, next is the termination. So the RNA polymer is already transcribed, this one already transcribed, and then it is found in the terminator box, so where the row factor is waiting for it. Thus as soon as entered the terminator box, the row factor attaches to the RNA polymerase, and thus it it then moves towards this one here, and thus it start breaking the hydrogen bond that form between this one. Okay, so what happened, as soon as the RNA polymerase reaches a terminator box, the row factor is waiting there, binds to the RNA polymerase, and then it comes back at the initial position and start breaking the hydrogen bond between them that is formed. See, I have written start breaking hydrogen bond. See, this is the elaboration. See, this is the RNA DNA strand, and this is the transcribe strand. See, as soon as the RP reaches a terminator box, what happens, the RP with the row factor comes like this and start from the initial phase by breaking. See, here he starts seeing the breaking, start breaking the hydrogen bond. See, it is breaking here. I am breaking this hydrogen bond, and thus the row factor is start breaking like this. It's breaking here. It will open like this. Our row factor separating. So the sigma factor is also separating the hydrogen bond. The row factor is also separating the hydrogen bond from the template, and thus the new strand is formed. The transcribed strand is mRNA is formed. That's the transcription process. Okay. Now by the process of the transcription, obviously there are different types of RNA are producing. So there are four types of RNA. Okay, means obviously you know that the DNA, after DNA the replication is also occurring, and the one of the segment is getting transcribed. So after transcription, not only the mRNA is only producing, there are different types of RNA that will be produced. So there is rRNA, mRNA, tRNA, and snRNA. mRNA is the one that will get translated and converted to the protein. Okay, but there are many other RNA. There are four RNAs. Okay, so the rRNA is called ribosomal RNA, mRNA is the messenger RNA, tRNA is called transfer RNA, and snRNA is called a small liquid RNA. You can just take the screenshot in here like this and can study this one, the difference between all of the RNAs. That is very important actually to learn. Okay, So just basically this one you have to remember, the massive messenger RNA is okay, massive RNA. And why have written the massive RNA? Because its size is much bigger. Okay, the nuclear sequence is much bigger, it is the biggest RNA. That's why I have compared it like this: messenger M for messenger RNA, M for my massive RNA, the biggest RNA. Just take this screenshot. If you don't can't understand, just
अरे
message
मेरी
me,
गलती
okay,
है,
it
मेरी
here.
गलती है, मेरी गलती है, मेरी गलती है, मैं बताऊँ क्या किया मैंने।
Here also
तो ये जो लिखा
the
है ना उसको
RNA
5 कर दो,
ribosome
मैंने
content.
3 लिखा है
You
वो।
can just take the screenshot
अरे
here and can
3
study. I don't have to I think don't have to remember about the
देखो
ribosome.